Great. Well, I'm really honored to be here. I'm a huge admirer of the Paul Allen Foundation. I think it's changing our understanding of who we are and increasingly tackling diseases and understanding why they happen in a way that no one else is approaching these problems. Really excited to moderate this. Special thanks to Catherine Richman, gracious introduction and allowing me to be here with all of you. I realize that the audience here is a little mixed, so I'm going to try to talk about where I think we are today. None of the panelists are thinking about the traditional approaches that I'm going to discuss, but I think we're going to have a lot of questions back and forth about whether what they're doing is completely novel in terms of mechanisms or whether it's going to feed back into the traditional pathways. So very briefly, dementia is a cognitive or behavioral disorder that interferes with usual function. It's a decline from previous levels, not explained by psychiatric diseases or confusion. It is measured by history taking from the patient, a knowledgeable informant, objective cognitive testing. It's one of the major set of diseases in the United States, the most costly illness from Medicare analyses, at least $200 billion a year. And unlike cancer and other disorders, the prevalence is increasing. We think this will double in 20 years in United States and other similar countries, but in middle and low income countries, the prevalence will triple. So this is really a huge worldwide problem. Want to say that this is a pretty new field. I think that we were described 4 major pathological types of dementia around 1900 by great neuropsychiatrists. But understanding the molecules associated with these different diseases really has started in the 1980s with Stan Prusner delineating the structure of the prion, George Glenner the structure of amyloid beta, and slowly different proteins associated with these degenerative diseases have been delineated. One of the fascinating aspects of this is that depending on which of these molecules aggregates in the brain, you see very different clinical syndromes, and that's because these molecules attack very different circuits in the brain. Touch on that in just a second. I think that for a lot of the biology that goes on with Alzheimer's disease, it was like cancer in the 1970s. We think about this as one disorder. And in fact, we now know that if you have Alzheimer's disease and you're over the age of 80, you almost never have a single Alzheimer pathology, A beta and tau. Even some of the genetic forms of these disorders can cause multiple proteinopathies. So the model, and I really attribute Stan Prusner to a lot of the thinking around this, is that we have degenerative diseases that are genetic or sporadic. Dan Geschwin has taught me a lot about the genetic forms of these diseases. The wonder of the genetic forms is they can be modeled, and we'll talk about the models in just a minute or two with this incredible panel. In all of these systems, we have 3 phases: preclinical, early symptomatic, symptomatic. Symptomatic is when we can recognize this at the bedside. I think increasingly we want to recognize these diseases in the very earliest stages in the pre-symptomatic phase. The degenerative diseases are caused by abnormal protein aggregation. Theme is how are we going to get rid of those proteins or prevent them from aggregating. And I think with many of these diseases, the proof is increasingly stronger that these bad proteins spread from one cell to another along specific circuits. So this is, I think, an important paper that Bill Seeley wrote in 2009. He took these different degenerative diseases. Again, we know they all have different molecules associated with them. looked at the atrophy pattern, and indeed, they don't hit the brain diffusely. They attack very specific regions: Alzheimer's posterior, frontotemporal dementia, frontoinsular, temporal lobe variant, semantic dementia, anterior temporal, non-fluent aphasia, left frontoinsular. Entirely different brain regions. And Bill asked the question, why are these regions hit so reliably with the different degenerative diseases? And what he did is he took young people, measured a bold signal in the brain, drawing region of interest in the area where these diseases tend to begin. And the punchline was that these diseases all spread along a very specific circuit. So these circuits we know we are born with, they grow as we mature, and then depending on the molecule that attacks them, there is a progressive neurodegenerative condition. Another area, and I think the group here is particularly well poised to begin to think about this very question, is why do these different diseases hit very specific regions? And I think Bill Seeley in our program would argue that, at least in frontotemporal dementia, this probably begins in a single, very interesting phylogenetically new neuron called the von Economo neuron. Hasn't yet been made in cell culture, but I think it's going to, and Martin Kapman is thinking about this problem. So the punchline in frontotemporal dementia is that in this layer 5B of the frontoinsular cortex, this seems to be the earliest degenerative neuron, and spreads along first von Economo neurons and then begins to spread out into adjacent neurons in layer 5B. So this is an example of that, and again, modeling could be incredibly interesting about why here, why this particular type of neuron. You can see in the first piece of this that the TDP43 protein is intranuclear in a normal neuron. Very early, and this is a very early patient, it begins to spread out of the nucleus into the cytoplasm. So the question is, we have 4 genes, big genes that cause frontotemporal dementia, and the question is, are these completely different molecular disorders, or is there something about the region, the von Economo neuron, that is telling us why the disease begins there? One of Dan Geschwin's fellows just presented data that I thought was extraordinary, that suggested that there was specific up and down regulation of gene complexes in frontotemporal dementia, whether it was caused by a tau mutation, progranulin mutation. So I think, again, with modeling, we can really think about selective vulnerability. With selective vulnerability, one of the interesting things that we have learned is that sometimes these diseases begin early with strengths that emerge in the setting of the disorder. So we've written quite a bit about the exceptionalism in art associated with the frontotemporal dementias, almost always left frontoinsular or left anterior temporal. But the majority of people don't develop this explosion of interest in art. So this particular piece is called Unraveling Ravel. It was done by Ann Adams. What she did was, before she was sick by about a year, she drew out the piece Bolero. She took all of the meters in the piece, chose her favorite note, gave it a specific color, showed the repetition of the note throughout this very repetitive piece by Ravel, and shows the change in notes in meter 323 with these gaudy fluorescent pinks. So the beginning of the disease is this extraordinary enhancement of visual creativity in her. Okay. So just a couple more comments. These diseases, I think, were really beautifully described by the poet Emily Dickinson, who many poems have a very relevance to neuroscience. So she was interested in moral decay, and this is how she describes moral decay. Crumbling, and I think you can just replace crumbling with dementia, is not an instant act, a fundamental pause, dilapidations, processes, or organized decay. And that is the circuit-specific, molecule-specific degeneration of circuits. 'Tis a cobweb. I'm picturing the first misfolded molecule in a von Economo neuron, a cuticle of dust, a bore in the axis, and elemental rust. Rune is formal, devil's work, consecutive and slow, fail in an instant, no man did, slipping is Crash's law. So that's these diseases, and I think the people in this room are as likely to solve them as any group I've ever seen. Just a last comment or two about where we are with the amyloid hypothesis. Rusty Gage and I were talking about this. I'm not an amyloid hypothesis person, and I think this hypothesis is failing. But we can diagnose this now. So I think with amyloid imaging, you can see that there is a massive uptake of this PIB tracer that binds to A beta 42. This happens probably 10 to 15 years before someone becomes symptomatic. So we really have a window into treating people very early in the course of the illness. It differentiates other dementias, and then the latest molecule on the block is molecular imaging of tau. I think this is showing us a different story, but also incredibly powerful for thinking about Alzheimer's disease. This is my last slide, and it just shows the spread of tau beginning in the medial temporal lobe into posterior temporal parietal regions, and then diffusely across the brain. It happens a little later than amyloid, but importantly, I think the tau is a marker for how sick neurons are. I can tell you at the bedside what that tau image is going to look like because it reflects dysfunction. So I think that's it. Lots of amyloid and tau therapies going on right now. This will happen whether any of you do anything. This field is set. The pharmaceutical industry is interested in it and will continue to pursue it. But I think we're going to need much more. And that's really what we're going to talk about today on our panel. So thank you. Okay. We have some amazing people here, some of whom I've met, others I haven't. Jeff Iliff, who thinks about lympholymphatics, and he's from Oregon Health Science University. We have Thara Karadotir from Cambridge. I didn't pronounce that well. Rusty Gage from the Salk, longtime friend. Amy Gao at UCSF. Martin Cabot, Michael Kaiser, and David Kokel, all from UCSF. Thank you. Wow. Amazing. Okay, I think the first question that I wanted to ask, and all of the panelists have thought a little bit about this, and it's really at the center of, of what they're all doing, and that is the traditional ideas that amyloid and tau are the cause for Alzheimer's disease. Amyloid aggregation triggers tau aggregation. And you're all thinking about that. And I guess one of the big questions is are the mechanisms that you are exploring these diseases working through tau and amyloid, or are you onto a very, very different story? I might ask Rusty to start, because I think he would argue almost certainly a very different story that he's thinking about.
Well, I don't think I would, I'm not against either of those hypotheses. To jump into it, I think that we need to move earlier than when the pathologies are present and look at stages of the disease, even pre-symptomatically, if we can get there. I mean, our bias would be that the primary driving correlate of Alzheimer's disease is in fact aging. And we need to understand the aging process as it leads prior to or in conjunction with the emergence of the syndrome. So we're interested in those mechanisms that are at the interface between aging. And then in particular, individual differences of how individuals age and correlating that with the onsets of the disease.
One comment about this, and I looked at the poster with Rusty and was really intrigued by some things that are Alzheimer's specific and some that are more age related. And my teacher, Frank Benson, always told me that I should study early onset diseases because they aren't confounded by aging.
Right.
So I think we have focused a lot on cohorts that are very tightly understood genetically who are young and don't have necessarily aging as an added
right.
kind of component.
I might comment a little bit about that because I actually trained in an aging laboratory since I was a postdoc with Cynthia Kenyon. And it's an incredibly powerful thing just to think about aging as something that could be genetically regulated. I don't think that's surprising to us now, but it was 10, 20 years ago. Then it brings up a whole other set of problems, though, right? Because the aging field, like every field, is filled with things that are absolutely true, things that are true under certain circumstances, and other things that are artifacts that somebody happened to get published. But work that's been very influential for us has been Dan Gottschling's work on yeast replicative aging and the effects of aging on the function of protein homeostasis, maintenance, I guess, machines in particular. Obviously the proteasome is important. There may be mitochondrial protease systems that are important, but he is focused on the lysosomes and the lysosome autophagy system. And specifically, the lysosome requires a very acidic pH to work. You all have heard this before, but the lysosome is filled with acid hydrolases. There are enzymes that require pHs under 5 in order to reach their optimal activity. These are dangerous molecules. You can't let them loose in a cell. Oh, you can't let them loose. I sound a lot better, or at least a lot louder. You can't let them loose in a cell, and so you have to sequester them in these specific organelles. And with aging, what happens is the lysosome becomes less acidic, and so overall proteostasis is going to be challenged as we get older. And I think there's a dynamic range for the lysosome in terms of its ability to respond to stress. And I think the ability of the lysosome and the proteostasis mechanisms to respond to stress potentially becomes worse and also narrower with age. And I think that's a really interesting area to explore.
And are you and Dr. Iliff working in very different systems, but both thinking about getting rid of bad amyloid and bad tau, or do you think almost certainly, and Rusty would argue this is much more complicated than those two proteins?
Yeah, I think that I would echo Rusty's comment that earlier is probably more important. I think from the perspective of how much we think about amyloid beta and how much we think about tau, the answer we give in terms of science and the answer that we give in terms of grants and publication are different answers. I think the work that we've done on the lymphatic system, the work that is presently being done on the meningeal lymphatic system is focused on amyloid beta homeostasis and its potential, the impairment of those pathways' role in dyshomeostasis, mostly because that's a narrative that people can grab onto. I think most of us, though, think in the conversations you would have over a glass of wine or informally, is that these are the changes in the glial compartment, the changes in the endothelial cell or the lymphatic endothelial cell compartment are probably very, very early changes that are going to influence astrocytic support of oligodendrocytes, right? Astrocytic support of neurons, astrocytic support of all of the different homeostatic functions that they're doing. Now, one of the consequences of that, one of the downstream consequences may be amyloid aggregation, may be the propagation of tau pathology, but there are also a number of knock-on biological effects that may underlie the disease state that amyloid pathology is simply a state marker of. So I think looking earlier at how that function, how that glial function, how those other cell types are disrupted both by aging and by the particular manifestation of aging that exists in the disease, right? So what differentiates an aging person from an aging person who is destined to get Alzheimer's disease is a question that we need to answer.
Martin, any thoughts about this argument?
Yes, I mean, I think, you know, I don't think we want to throw out everything that, you know, over many years has been studied with respect to tau, A beta, and as you pointed out, many other proteins that aggregate and that might propagate in a prion-like manner in these diseases. Because the good thing about these mechanisms, or the very interesting thing, is that there seems to be a common theme connecting different neurodegenerative diseases. And at the same time, some specificity as to how they play out, and you brought up the exciting topic of selective vulnerability. So I think by both learning from common mechanisms that might be common theme between neurodegeneration, looking at the convergence of the human genetics, the mutations in those pathways, as well as the protein pathology, I think we can learn something about some general mechanisms, and then being very alert and using all the newest technologies, both in patient brains as well as in vitro models, to understand selective vulnerability mechanisms, I think is probably a very fruitful approach. And our group has been focused on prion-like propagation in tau, which we think is an important common mechanism. But I would also say that I think human genetics is an extremely powerful way to discover in a completely unbiased way other pathways that might be important. And as we have been able to sequence deeper and through GWAS studies discover so many other hints for possibly important pathways, I think we need to broaden our view of what might all play a role, right? And so I think listening to the human genetics, realizing that, and Jeff mentioned this, that genes acting in very different cell types and even systems in the body might play a role based on the human genetics, and having a good way to address that both in the human patients as early stages of the disease, so having some ways to look longitudinally how early changes occur, and then being able to model it in systems that are amenable to experimental perturbation, so where we can really test causality, and then having a good integration of going back and forth between in vitro lab models and patients to try to understand how the human genetics play out mechanistically and how we could intervene in therapy.
Great. I want Michael and David to have a chance to comment on this. I think that's a really good, set of comments to move to the big question I want you all to address also is, where does this disease begin? What's the most important? Thora, is it vascular? Is it white matter? Amy's excited about glia. But before we move to that, would you guys like to?
Yeah, sure.
Comment?
I mean, you mentioned these early onset forms, and I think if nothing else, using those genes as triggers to model the disease in animals will be incredibly useful. And also having animal models that scale, that we're going to all be generating lists of genes and hopefully drugs and compounds that we want to test against these phenotypes. And I think to do that, having a rapid cycle, having a way to a model that it's easy to take shots on goal, a rapidly aging model would be useful to develop those.
Great.
Well, and also just to pick up on Dave's point and some of the other points we've heard, and bring in a small molecule perspective, because of course, if you're coming with a system where you know that the molecules get in and they induce a phenotype, they modify an endpoint that we care about, then we at least have the advantage there that the question becomes not whether we could find a molecule that would work, but out of a collection of molecules that have some degree of effect, why are they working? And even if we're talking about a system, and this is some of the work that we talked about at our poster, a system where we purposely pushed the model onto the knife's edge, where we're looking at something like tau and the prion-like propagation of tau between one cell and the other, and it's in a case where tau is upregulated. So we know, in this case, that tau is more present than it would otherwise be. The neat thing is you can get at some machinery there. You can get at machinery that is either being stressed by the situation that you've pushed that far. But then if you find molecules that help push you back over the knife's edge on that machinery could indeed trace back to some of these other questions we've talked about. And so that's one hope we have as a way to contribute to this, because then you can take those molecular agents and those hypotheses of machinery that come from that and feed it back and see if, for instance, that helps earlier. And I think a neat thing about this, and a way it complements, is that it doesn't rely on tau itself being the cause. It could be that tau is the lion that you let loose, that this machinery usually controlled it and other things, and that letting loose the lion is not good news for the city. But you wouldn't have even let the lion loose originally if the machinery had been in better shape. And so we're trying to find that point. So that's just a perspective I wanted to toss into the ring with this, to see if that might fit into some of these other hypotheses.
I want to make one comment that, which I think is very Paul Allen Institute specific. And I think it's been brought up already, is this idea that really important to think about human tissue and understand what's going on. And I'm hearing this echoed across this group. And then the other aspect, is technology. And, we can see the amyloid and tau, but now we can, simultaneously with new techniques, image 35, 50 proteins in an individual neuron. And maybe, like Michael says, maybe tau isn't the first or the third or the fifth molecule. And, I think that there is so little known about these diseases when we get down to the pathology that having a very basic group like this focused on anatomy, mechanism, cell models is just unbelievably powerful. Thora, where does this... You wanted to say something, Amy, before we get to Thora? Yeah.
Yeah, thinking about amyloid and tau and where diseases start and what cells are important. I think there's an analogy to be made, and I think, we owe our ability to have organized and understood these diseases entirely to neuropathologists who identified these important molecules, and then cohorts could be organized, and then we could find the genes. But if we use the analogy of, let's say there are 6 serial killers on the loose, and each of them, one person kills with a bloody knife and throws it in the trash can. Another person uses, I don't know, something else horrible and throws that in the trash can. I think at this point, we need to stop digging around in the trash can, and apologies to Bill Seeley, because the crime's been committed. The crime is in the house. We need to figure out how the serial killer got in. And they're not all the same killer, and they use different mechanisms to get in. So we need to think hard about way upstream, just like Rusty was saying. As a readout, as a way to organize our thinking, even as biomarkers, what's in the garbage can is going to be helpful. But I think if we want to intervene on these diseases early, like Bart de Struber was saying this morning, we need to move way, way, way upstream from the protein aggregates that we can see.
Great. So, Thora, a little bit about where these diseases begin. I guess we're going to talk about Alzheimer's first. Just a comment. I think when our pathologist looks at the brains of elders and very old, over 85, that's older than me, the vascular pathology becomes very important and evident and is a cofactor in the majority of cases. Does this begin in other forms of Alzheimer's, early age of onset? What is the critical cell for this aspect of these diseases?
I think I would be somewhere else if I knew the answer to this question. We are working on a hypothesis, of course, that there is some vascular component to the disease in general. And just as any machine, we need food and energy. And what is known about in the white matter is that is the most vulnerable place, subcortical white matter in the brain. And we have absolutely zero understanding about the vasculature in that. And we know that the vasculature with aging gets worse. We know that majority of people, as we age, we start to have cardiovascular problems. And we also know that there's a link to now in the Western societies with a better control of cardiovascular disease. actually the incidences of AD or Alzheimer's disease is lowering. So there's a good correlation that this may be some kind of underlying factor. Just generic health, like the Roman says, I'm not going to say it in Latin, a healthy brain in a healthy body. And then we are looking into the fact if the oligodendrocytes or the myelin is getting destroyed in the subcortical white matter, there's a lack due to lack of oxygen supply, and whether that affects the ability of the neurons to carry out their job, and then cumulatively with other factors that will lead to a degeneration. But that was very upstream, and hopefully if we have good MRI scanners and we would put a lot of money into healthcare and to scan people on regular intervals, just like you do with cancer screening, we could be able to pick it up early. That would be a nice dream.
But I think the question itself speaks to where we're at, right? So if we have serial killers wandering around our neighborhoods, I'm not sure where we are at in the metaphor right now, but at the end of the day, I think most everyone's agreed that the point of leverage and the point of change where the aging brain that is going to age in a healthy manner versus that which is going to age in an unhealthy manner, those are probably diverging decades and decades before the onset of perhaps even amyloid deposition that's detectable by PET, right? So maybe even before then. So the question isn't so much, what are the cell types? Do we see tangles in neurons? Which neurons? Do we see tangles maybe even in astrocytes in some of these disease states, right? But what's changing 30 years ago? What cell type is that? And I think most of us have a PET cell. I believe that the astrocyte is the seat of human consciousness, and that's actually where the soul resides. And so I'm sure it's the astrocyte, or it's the oligodendrocyte, or it's the neuron. It's, Betzos-Lokovich believes it's the pericyte or the endothelial cell. But at the end of the day, we just don't know. And I think actually one of the things that the Allen Foundation and the Allen Brain Institute has exemplified this is a commitment to understanding all of the diversity of those cell types and how they're actually changing in that state and how those changes predict what's going to happen. And I think when we start to understand, what about choroidal epithelial cells or ependymal cells or stem cells or these different cell populations in that state, that's where we can start to answer that real question in a way that isn't just us, taking a guess at our favorite cell type,
Go ahead, and I'm going to ask Rusty a question too, please.
Just one interesting bit of information and again, I for sure don't think that, for example, tau aggregation spreading is the only mechanism, but Braque has seen tau aggregates in many decades in the brainstem before it occurs in higher regions in the hippocampus and entorhinal cortex and so on. So it's still possible that even protein aggregation in very specific brain regions is a very early event, and that was then causing the disease is the slower spreading to other regions. So it's possible that is one of these slow processes that starts very early on, but even after it starts, there might be a way to slow it down to prevent it from taking over other brain regions, possibly.
But I think that one of the diversities that is a little less understood in that space is, when you think about transentorhinal pathology that is present in a lot of people, right? Why is it that it spreads in some people and not in others? Right, so I think that the ubiquity of pathology in certain brain regions, right, in contrast to the generalization of that pathology in other subjects, those host differences, I think, may be the more important differences. What is it that is influencing the pervasiveness and the rate of that spread?
Yeah, a comment on that, and then I want to come back to the cell type. So, I think the Allen Institute, or foundation, has been a little agnostic about what all of this, genetic mapping is going to tell us. But I think going back to Jeff's point, I have no doubt that, and I think this is more used in our laboratories at UCSF than Wikipedia or Google. So, I know all of our scientists are constantly using the Allen map, and I am quite sure that these factors are incredibly diverse and vary from person to person. And the more we understand of human phenotype, the better we will be able to understand this.
So, and related to that would be, we focus on a common theme of a common disease, and I think there's an encouragement now to really look at diversity, and that not just the different types of AD that there are, but that individuals are really quite different, and in particular how we age as individuals and how our individual organs may age at different rates even within an individual person. And understanding and appreciating and sort of embracing the differences is important. And one way to think about that in studies going forward is in stratifying patients, populations, so that you working together with the clinicians and the basic scientists so that the samples that we're working on really reflect a range, if not extremes, so that we can get insights into how some patients may be resistant to even the same mutations as those that are very vulnerable to it. And this may give us clues in both mechanism, but also therapeutic interventions as well.
I want you to comment specifically on your poster. So you showed these changes in glycolysis in neurons, and I haven't got much traction on this question. What is the cell to study that'll give us the best understanding of the disease? Do you think you see similar things in other cells, or is this neuron specific?
Well, that particular study, just a little background for people who didn't do this, is just using a methodology, this induced pluripotency or direct reprogramming. In the induced pluripotency, what happens is, as promised, you reprogram the genome so the epigenetic marks are gone, so any of the age-related phenomena that you wanted to observe are erased. So when you're looking at age-related diseases, it compounds the problem of finding things. So by using direct reprogramming into neurons, you can capture and retain the epigenetic marks that are there, and you can find things that are there. Now, the unique thing about neurons with this regard, and I think why it was picked up here, is we focused on, in this particular study, on neurons and used a neural-specific study. And neurons are uniquely dependent upon oxidative phosphorylation. So when they shift from an early neural progenitor cell to a neuron, they actually shut off the glycolytic, or a lot of the glycolytic pathways. They switch isoforms, and it really becomes. So the remarkable thing, or the thing that you're picking up on the paper, or this study, is that in Alzheimer's disease, this is not an age-related event. This is a uniquely Alzheimer's event, or at least with regard to these patients, that there's an upregulation of, or a shift from oxidative phosphorylation to glycolysis, which of course for people that are in broader fields like cancer, hear this as a Warburg-type effect happening. And this is all with an inclusive downregulation in aging of mitochondrial function. So you have this downregulation of mitochondrial function, and with the inclusion of the disease phenotype or the disease genotype, you get this similar type of shift of increasing use of sources of energy from other places, and the consequences of switching to new energy sources are paramount.
And Michael, David, what are your favored cell systems to think about screening and pathogenesis?
Yeah, well, I guess, focusing on specific cells and breaking them down and culturing them is incredibly important. I also tend to think about the intact organism as being very important in, integrated systems. So we focus a lot of our attention on zebrafish, on intact brains, invertebrate brains, that we can get access to lots of different animals. And I think that's great because it combines the glia, the neurons, the vascular system, the microglia, all the systems that we know are involved in some way, but we don't really understand.
Great.
Well, my short answer, coming from a computer science and machine learning perspective, is my favorite systems are the one for which I have data that I can turn around and get experiments done in response to in a timely manner. And so as long as the data set is large enough, it's driven by an original hypothesis, because I will make that distinction. Big data is a big thing these days, but it should be focused. and it should be collected under a particular hypothesis that then we're coming back and attempting to interrogate. And the way we interrogate that is by working with close collaborators to actually get causative focused experiments done. So I feel very fortunate. There's a bunch of us in this room and up on the panel that I'm very delighted to collaborate with.
So we have a few minutes, Catherine, or 5? 5, okay. So I was going to ask you all about amyloid and tau and ask you to take a stand, but I don't think I will. I think Martin will take a stand very quickly, and then the last question. What's your stand here?
I think, just to summarize what I said, I do think we shouldn't neglect them because there's a lot of data, and certainly when you look at tau, there is a correlation of tau pathology and dysfunction, neuronal death. You can study it now with PET probes longitudinally. So I think there's a lot of great tools to continue looking in that direction. But I also think, as I said, human genetics also, if we just have an unbiased mind and look at what the new human genetics tell us, they also point in other directions. Certainly, vasculature and glial cells, and I think building equivalent system, and we're excited going forward to work with Eric Julian on his astrocyte neuron co-culture systems, brain organoid systems, to really dissect cell-specific contributions and find out in which cell types do specific genes act, and what are the molecular determinants of selective vulnerability. So I think that is a very fruitful direction to go back and forth between human genetics and then a system in which we can control these genes one at a time.
Fabulous. So where are we going to be in 5 years? That was the question, Thora. Maybe you could start us out. We're going to have much better interventions for vascular disease in certainly high socioeconomic status populations. Whether we can budge this in low and middle income. But where are we going to be in terms of, what are questions we'll be asking that are different than they are today?
5 years.
5 years.
Wow. In the short term of 5 years, well, I guess we can just be, I think we will be looking more into general of the brain. I think the start of we will be moving away from being neurocentric to be more whole cellular interaction physiology, understanding of how the brain works, to understand what is the basic function and how it changes with aging and how we can perturb that and understand when it gets wrong with either mutations or point mutation in some certain genes and proteins.
Great. Jeff or Rusty or Amy?
Well,
we'll
I would
start
just
on
say
that.
that we heard a great talk by Bart, and I am convinced that this morning that we're going to see a bunch of studies coming out over the next 10 years probably. They're already on their way. They're going to be there. So it's probably a little early to put these hypotheses to bed or to bury them, and it's got to be refined. But I also think that it probably is an opportune time to begin looking for broader questions about the biology, and it comes at the right time. The tools are here to do this that weren't here 10 years ago, and to apply the sort of things that we've heard today, some of the newer techniques, to these disease-related phenomena is going to be, I think, where the future will be.
Great.
So I'm an optimist, so I think in 5 years, Paul Allen will be announcing that he's setting up a shop where you can arrive with your brain. He'll dissolve it. You just bring your ECM of your brain, and with active genetics and Gestalt and memoir and bioelectricity, he'll grow a new brain for us, right? That would be the super, super optimist. I actually, I look to cancer for where we should go. I think the direction we need to head is the path that cancer took, which was very personalized, very mutation specific, and we are at the point where we have a lot of the genetics, not all of it, but a lot of the genetics to do this work thoughtfully, that's very sort of disease specific, but by disease I mean, an APP mutation is different from a presenilin mutation, is different from a MAPT mutation, is different from ApoE, Alzheimer's disease. So even though I am an optimist, I think we're 20 years behind cancer. We might be able to catch up in 10 years. That's the hope.
Great. Couldn't agree more. I am very dubious that 5 years from now people will be talking about an Alzheimer patient without knowing whole genome, having incredibly good biomarkers to verify whether that's true or not. I think we will have much deeper information sets to think about this. And in parallel, I think with the Allen programs, we will have much deeper data to think about what these diseases are doing in the brain.
Yeah, I think that, echoing that, I think that those two influence each other a lot. So as our human genetic understanding goes beyond GWAS findings to private mutations and very, very, very low frequency variants that end up contributing in a small incremental manner to the development of the disease. I think in 5 years our understanding of that will be different than it is now, and the consequence will be the identification of people that are getting to Alzheimer's disease through a glial or an inflammatory or more of a vascular route. And that changes both, I think, our clinical therapeutics perspective, because our clinical trial design changes and the identification of vulnerable populations and treatable populations changes, but it also changes how we address the biology. So instead of our animal models of Alzheimer's disease being overexpressing mutant presenilin-1, we are instead trying to pull out those subtle phenotypes and really get to the bottom of that biology. And I think that becomes there's a lot more leverage there.
Fabulous. And we have 1 minute. I think I want Michael to say he's going to have a small molecule for subtypes, and Martin and David are going to have the right cell systems to study in vivo, in vitro, and say a little bit about that.
Well, so one thing to echo this point about there being a fair amount of heterogeneity, I don't think it's going to be random and unbounded heterogeneity. I think one of the things we're going to find, and I don't know if it'll be 5 years, but one thing I think we are already suspecting is the case is that there's going to be lower hanging fruit and then harder to reach fruit in this. And I think one of the important steps we need to do is start delineating those different spaces, either from the perspective of small molecules or from a genetics perspective and others. Obviously, each of us are here with a particular motivating or guiding mechanism or hypothesis in mind, but we're beginning to carve off some of these spaces. And I think calling it Alzheimer's disease is a little, in a way, misleading in the same way that you can't say I'm going to treat cancer itself. There's a lot of roads to Rome, and there might be multiple Romes here that I think some are going to be easier than others to address.
Beautifully said. I think we have run out of time. Sorry, I'd love to hear this panel go on forever anyways.
Well, actually, we wanted to save some time at the end of the session to allow questions from the audience for the panel. So we have about 15 or so minutes, 15, 20 minutes. So I see Bart is ready to.
Yeah, I had difficulties to shut up a bit, because sometimes I feel that we are sticking a bit in the past. I remember the discussions about amyloid and tau, and the people who worked on tau said amyloid is wrong, and the people working on amyloid said tau is wrong. I think we should go beyond that. So I think there is truth in all these hypotheses. There is clinical evidence that it's important. The genetics clearly show that a mistake in amyloid is driving a small subgroup of the patients. And I just think we need to go beyond that. The therapies don't work. Well, we should have a look at the therapies, and we should focus it on those groups where we know that amyloid is really a problem and try to solve that, and then move to the next group of patients and see what's happening there. We know that inflammation is incredibly important. I didn't hear too much about that. So let's now solve the inflammation problem. Those people who have TREM2 mutations, and then people who have a certain genetic risk for inflammation, let's sort that out and see in patients which are on the way there, let's treat them with this type of drugs. And then in the end, you will have a combination of drugs, because I'm sure that also in the people which are mainly driven by a genetic inflammatory process, that the amyloid or the tau is playing a part. And in fact, we should much more look, we are so pessimistic in our field. You should have a look to for instance, cardiovascular disease. Nobody's discussing about the role of cholesterol, but it's in a familial disease, it's driven by cholesterol. In the sporadic form of the disease, cholesterol plays a part of it. So we think we need to think much more integrative. That's the first point I want to make. And the second point is about drugs. We are talking about small compounds, we're talking about antibodies. For me, the really exciting finding is the one which I mentioned this morning, is spinal muscular atrophy with antisense therapy, which for the first time gives us a rational way to treat molecular mechanisms. I expect, and I'm the optimist, if we do this well, if we organize this well, one of these neurodegenerative diseases will be treated with these antisense molecules in 5 years. It's possible. So that's the two comments I wanted to make.
I might make one. I love the panel. One part of what you said that I really agree with is that we will make much more progress if we focus on really well understood cohorts where we think we understand in a very precise way the mechanism. You know, I think studies of populations from 60 to 95 saying that this is all Alzheimer's disease and amyloid are doomed. I mean, I think absolutely doomed. So, yeah, comments?
Yeah, and just, underlining what you said, I think one of the things we'll learn in the next 5 years is if we have stratified clinical trials that are well executed, where we collect a lot of data, that's the ultimate experiment for mechanism, right? Because we're doing an experiment, an interventional experiment in the relevant model system, the human patient. And I think with some of these lowering approaches, and as you pointed out, I think, Bruce, stratifying the patients is very important, and having a high resolution readout on the phenotyping. That ultimately will teach us a lot about the basic mechanism as well, right? If it's well done.
I was hoping that we'd hear potential cells of origin that reside outside the brain, such as emerging evidence for a gut origin for Parkinson's disease based on not only microbiome distinctions, but epidemiologic data that vagotomy profoundly influences the risk for PD, or from GWAS that age-related macular degeneration. It's many of the same genetic variants and pathways. There's significant overlap with Alzheimer's, so there may even be an origin in the eye with retrograde ascension.
Want to comment on the microbiome? I just seen a really nice presentation. But anyways, go ahead.
Well, I agree that there are going to be cases, and in fact, we had a really interesting one recently. It was actually I think it was a TDP-43 case where probably the pathology started in the GI system. I feel like that it's unclear to me if that's a different disease, or it's the same disease, or there were genetic modifiers within that person that caused it to express in a slightly different way. I think they're important questions. I'm definitely a proponent of studying both the common diseases as well as some of the outliers, as long as we recognize that they're outliers, because they could teach us something about general mechanisms. Beyond that, I cannot comment because I'm not an expert in Parkinson's.
Other panel members want to say a little bit about the microbiome? Well, I mean, I think the group at Caltech, for one, is Tim Sampson, has very nice emerging data around the idea that maybe E. coli is particularly toxic in Parkinson's disease, and I think it's a target. Whether this will be a major target or a minor target or not work at all, I think we don't know yet. One of the interesting thing about Tim's work is that he's showing that there are proteins that are produced by these bacteria that might seed neurodegeneration in the gut, which is probably where Parkinson's disease begins.
So,
Dan?
Yeah. I was. There were kind of two themes, two of many themes that I heard. One of them is this notion of early detection, that this is a long-standing disorder, that these disorders proceed, or syndrome that proceeds slowly over time, and when we get a pathologic specimen, it's clearly at the end of the stage. A lot has happened. The second piece is this cell type vulnerability. And in thinking about it, as we're profiling, let's say, Alzheimer's brain or frontotemporal dementia brain, we're profiling the cells that have survived actually decades of disorder. We're actually not profiling those that were most vulnerable because those are gone, probably. And so I'm wondering, so it makes me think about these technologies that people are developing for single cell analysis, some very fancy, especially genomic tools that one could think about identifying cells that are going to, in 5 years or so, or the very, very earliest stage. Is there a way to begin to be able to predict that? And even start with a random labeling strategy, for example, and then kind of use that type of information. Because what I'm concerned about is that in thinking about this, the interpretation of the data that we get from single cell sequencing will be, we may not quite be able to interpret it because we're looking at those cells that have survived, not those that have actually died. Anyway, just wanted to throw that out there.
Yeah, I think that's a really good point. And again, looking at cells, not just their transcriptional profiles, but also their morphology and their physiology. I mean, in Parkinson's disease, for example, the substantia nigra is unique in a lot of ways, especially the dopamine-containing neurons in terms of their firing rates, their really high dependence on mitochondrial function. Because this firing rate, they're an enormous branching with such a small neuron to have all these branches out there. They're particularly vulnerable in their activity based on their high activity. So I think really, to your point, really amplifying on understanding the nature of the individual cells and why they would be vulnerable, not just at the end stage, but even as a consequence of their lifespan.
So I think it's a great point also, and Bill Seeley and Matt Jacobson, Xiaopan Xu, and Michael Grabe and I have been thinking hard about this, thinking about comparing vulnerable and invulnerable cell types, because we do actually know for most of these diseases, the common forms albeit, where the particular cell types that this starts in. And so I think it's a nice way to interface with the Allen Brain Institute, because they actually have transcriptional profiles for many of these cell types already. And then we just have to do the experiment. So if we put in a particular mutation that gives Alzheimer's disease in cells that come from the hippocampus, will that make those cells vulnerable, but the VENs are invulnerable? And then we can start dissecting out the differences using all kinds of novel tools as well as computational processes. I think it's a great point.
I think the same approaches can be taken not for vulnerability, but culpability, right? So if there are selected groups of neurons that are vulnerable, there's probably bystanders that are responsible for the crime. And I think one of the things that we're learning is that a microglial cell in one part of the brain is not the same as a microglial cell in another part of the brain. And this is also true of astrocytes, right? So there is a spatial and a temporal and a developmental heterogeneity that probably also interacts with the aging process and the onset of disease. Microglial cells associated with plaques look different and probably act different than a normal cell. So when we're looking for who's the problem, right, not just who's the victim, but I think a spatial and single cell understanding that's now possible is going to help that a lot.
Hi. My name is Andy Wyrobic. I work at the Lawrence Berkeley Labs, and I'm funded by NASA to see what might happen to astronauts, young male to middle-aged male, receiving a bit of space radiation. What are their downstream risks? And I wanted to share with you some of the concepts, one or two concepts that we're seeing. One is that when you radiate these animals, low doses, that doesn't reduce their lifespan, and they, as a group, have small behavioral changes, but nothing significant. What we notice in almost all the animals is like 2 or 3 months after exposure, we look at the cerebral spinal fluid, and we see changes at the omics level, proteomics, that are associated with vascular damage, with immune dysfunction, with glucose metabolism just way down. One of the sets of proteins just points to the choroid plexus, saying there's something wrong. But when we look histologically, except for the choroid plexus, we don't see anything. The vascular damage we don't see until like 9 months later. The amyloid abnormalities we don't see till 9 months later. So the rat lives 2 years. So it's towards the last third of the lifetime of the rat after being exposed early in life. So I'm asking the question of this sort of risk, which suggests it's sort of like an accelerated process of some sort. So we see amyloid, we see vascular fibrosis. That's one concept. The other concept, which I haven't heard, and I don't know if it's buried within your work, is the concept of microenvironment. Is the formation of abnormal proteins and needing to get rid of them is a normal process in biology. I mean, especially in the brain. So the scavenging processes, call them what you like, that need to get rid of these things, are they the culprit? In other words, this normal buildup, but if you don't wash it away at night, and over the last year or two, there's been at the Society of Neurosciences tremendous, and elsewhere, there's tremendous interest in cerebral spinal fluid and sleep, and that this is a time when everything gets washed away. Are these important mechanisms in terms of what you're talking about?
Yes.
Glymphatic.
So,
Please.
but we don't know. Okay, so there's a clear clinical association with sleep disruption and these downstream processes. How that works, whether it's a metabolic answer, which is the hypothesis that the group at Washington University and David Holtzman sort of believes, which is sort of a depletion of a less energy intensive slow wave piece, or whether there's a synaptic answer to it, or whether it's the glymphatic exchange of the interstitial compartment. Those are biological hypotheses that we have some decent answers to in animal models, but we don't yet have answers to in human models. The imaging of glymphatic function during sleep is only just starting to get underway. The project that we have that's funded by the Allen Family Foundation is to develop non-invasive approaches to do this between sleep and wake, and we've made some progress. But it's a pretty big step between that and being able to causally implicate it in these downstream processes. On a related note, if you are planning on sending people into space for long periods of time, one thing I would worry about, not just radiation, is actually the influence of microgravity, right? So the influence of microgravity on CSF circulation and the processes that are dependent on CSF circulation might be an additional concern that people who are sending, especially young humans, to Mars or things like that might consider. But I think the jury's out on how important that is in real human disease, but a lot of us are thinking really hard about it.
Dora, thoughts about this vascular etiology and the trigger that sort of sets a cascade going in white matter? And,
Well, that's really our hypothesis, that there may be some vulnerability in the fact that if you have, maybe with going into the gravitational force that you're going up if you're being an astronaut, may also have a mechanistic effect for the microvascular damage. But what we see also with, there's an interplay with glial cells. So also with aging, glial cells, I forgot to say that earlier, show much accelerated aging before neurons. So there might be their response to either lack of oxygen or lack of glucose, or with mechanical injury, and particularly culprit microglial cells can go into a vicious circle and then kind of circles around and can cause damage to the neuron, so that becomes the victim, like Jeff just described. So,
if I could, jump in on the protein level, so proteinopathies, right? And the serial killer metaphor, you know, how good has the forensics been about the protein aggregates themselves? I always think about proteomics as determining the proteins with complete molecular composition and not inferring that from what I call bottom-up proteomics. And so if the forensics isn't up to snuff, is there value in determining that atom composition of fibrils, and then, as the gentleman in the middle, walk it back from mechanistic points of view? I just wonder how excited the panel could get about really good proteomics.
I love the question, and I think it's really phrased in a smart way. And the truth is, I think we've analyzed and determined what was analyzable and determinable. The technology's changed, and I think a completely new look at the proteomics of this is going to be really important.
Isn't part of the problem that we're looking at the very end stage and trying to infer what is causally happening. So getting the material to do the proteomics, I don't know where you went. Oh, there it is. I refer that to the pathologist, but this is part of the issue is that quality of the material to do the kinds of work that you want to do. And then that takes us into the whole area of animal models that replicate the system well enough to extract the tissue, which we haven't sampled here in terms of animal models. But so that's at the balance between those two issues, at least.
Great.
We have time for one more questions?
Sorry. So just to add up the next 5 years onto the where it will be, and just as a comment is, it will be more, is there any new tools or the chemical tools or the biological tools to apply on a retrospective studies I work with, and is there any way of mechanism? Most of the clinical trials already done on phase 3, and because you showed the very nice pictures on the Biogen aducanumab of the Solanzimab. Not only that, is there any way we can access those samples, more focus on those clinical trial subjects, quickly understand much faster in a retrospective way rather than a prospective way? So that may be a place to start. And any tool, is it the cell type or the protein aggregation or the antibody, any techniques what we have, focus on just that clinical trial subject and subtype them. It could be a simple inflammation assay. So in strictly next 5 years focus, can we use those sample sets available for mostly the research community? So just a comment last question. Is there any way we can access those clinical samples to the research community?
I'll start. I think this is, well, this is controversial. How's that? So I think pharmaceutical companies historically have been very reluctant to share samples, and I think there are many examples of this. I think increasingly it's going to be very important that scientists become ferociously tough about making sure that when they participate in a clinical trial, there are agreements around papers, sort of data that will be shared so that these things don't end up in a dark hole. Really important question, I think.
Thank you.
Okay, well, I think our panel, a hand of applause.